Hello sir, this is my laboratory 2. Kathleen Rose F. Labog from BS Ed. My second topic is the process of mitosis and meiosis. What is mitosis? Mitosis is a process of nuclear division in eukaryotic cells that occurs when a parent cell divides to produce two identical daughter cells. When we say mitosis is a part of the cell cycle in which replicated chromosomes are separated into two new nuclei. Mitosis is conventionally divided into 5 stages, known as prophase, prometaphase, metaphase, anaphase, and telophase. The first is prophase. During prophase, the replicated pairs of chromosomes condense and compact themselves. The pairs of chromosomes that have been replicated are called sister chromatids, and they remain joined at a central point called the centromere. A large structure called the mitotic spindle also forms from long proteins called microtubules on each side or pole of the cell. Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. The next is prometaphase. Prometaphase is the second phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prometaphase, the nuclear envelope that encloses the nucleus breaks down, and the nucleus is no longer separated from the cytoplasm. Protein formations called kinetochores form around the centromere. The mitotic spindle extends from the poles and attaches to the kinetochores. The next is metaphase. During metaphase, the microtubules pull the sister chromatids back and forth until they align in a plane, called the equatorial plane, along the center of the cell. When we say metaphase is a stage of mitosis in the eukaryotic cell cycle in which chromosomes are their second most condensed and coiled stage. The next is anaphase. Anaphase is a stage of mitosis after the process of metaphase, when replicated chromosomes are split and the newly copied chromosomes are moved to opposite poles of the cell. During anaphase, the sister chromatids are separated simultaneously at their centromeres. The separated chromosomes are then pulled by the spindle to opposite poles of the cell. Anaphase ensures that each daughter cell receives an identical set of chromosomes. Telophase. Finally, during the telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. The chromosomes begin to uncoil, which makes them diffuse and less compact. Along with telophase, the cell undergoes a separate process called cytokinesis that divides the cytoplasm of the parental cell into two daughter cells. Telophase is the fifth and final phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. What is meiosis? Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half and produces 4 gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. During reproduction, when the sperm and egg unite to form a single cell, the number of chromosomes is restored in the offspring. Meiosis begins with a parent cell that is diploid, meaning it has 2 copies of each chromosome. The parent cell undergoes 1 round of DNA replication followed by 2 separate cycles of nuclear division. The process results in 4 daughter cells that are haploid, which means they contain half the number of chromosomes of the diploid parent cell. Meiosis is a special type of cell division of germ cells in sexually reproducing organisms used to produce the gametes such as sperm or egg cells. The process is split into meiosis I and meiosis II, and both meiotic divisions have multiple phases. Meiosis I is a type of cell division unique to germ cells, while meiosis II is similar to mitosis. Meiosis I, the first meiotic division, begins with Prophase I. During Prophase I, the complex of DNA and protein known as chromatin condenses to form chromosomes. The pairs of replicated chromosomes are known as sister chromatids, and they remain joined at a central point called the centromere. A large structure called the meiotic spindle also forms from long proteins called microtubules on each side, or pole, of the cell. Between Prophase I and Metaphase I, the pairs of homologous chromosomes from tetrads within the tetrad, any pair of chromatid arms can overlap and fuse in a process called crossing over or recombination. Recombination is a process that breaks, recombines, and rejoins sections of DNA to produce new combinations of genes. In Metaphase I, the homologous pairs of chromosomes align on either side of the equatorial plate. The last is, in Anaphase I, the spindle fiber contract and pull the homologous pairs, each with two chromatids away from each other and toward each pole of the cell. That's all. Thank you.